The work of Nobel Prize has transformed our understanding of the world. While approaching the Nobel Prize winners in chemistry, we not only learn their achievements, but also the spirit of honesty, tenacity, innovation, and forging ahead.
Hello everyone, welcome to today's class, the Nobel winner in chemistry. 同学们，你们好，欢迎走进今天的课堂，化学诺贝尔奖得主。今天会给大家介绍一位失去左手臂的诺奖得主萨姆纳。Today I will introduce you a Nobel Prize winner who has lost his left arm. He is James Sumner. Physical disability and defects can afflict people's human body and nervous system, but they cannot eliminate human will and the spirit of devoting to science. James B. Sumner, who had lost his left arm, won the Nobel Prize in Chemistry in 1946 for purifying and separating urases crystals. He's the convincing example. James B. Sumner was born in 1887 in USA and worked at Cornell University. At that time, he won the Nobel Prize. His prize motivation was for his discovery that enzymes can be crystallized. James Sumner shared the Nobel Prize in chemistry with John Northrop and William Stanley. He was the son of Welsh cotton manufacturer, learned early in life how to rise to a challenge. But when he was 17, Sumner accidentally shot by a hunting partner and lost his left arm. Although he had originally been left-handed, the young man retrained himself so well that he can continue to excel in sports, becoming a champion tennis player and a capable skier and mountain climber. However, friends and teachers warned Sumner against trying to follow his dream, becoming a chemist. Ignoring the warning, Sumner persisted his dream. He graduated in chemistry at Harvard University. In addition, he returned to Harvard as a graduate student in biochemistry. After receiving his PhD degree, he joined the faculty of Cornell Medical College, where he remained until his retirement. Sumner was the first one to isolate and crystallize an enzyme that showed it to be a protein. His work helped to stimulate research in enzymes and viruses. His research on enzyme urease, which is found in jack beans, as we show in the picture here, laid the foundation for subsequent work by others that elucidated the crystal structures of a large number of biological macromolecules. At the end of the 17th century, it became clear that biochemical processes don't necessarily require living cells, but are driven by special substances, enzymes, formed in cells. James Sumner studied the enzyme urease, which breaks down urine to ammonia and carbon dioxide. In 1926, he succeeded in isolating crystals with high activity from jack bean. Following research indicated that the crystals were composed of a pure form of urease and that the urease is a protein. Sumner's crystals were clearly protein, and however, when he tried to separate the protein from them, they always failed. He was therefore forced to conclude that the urease, an enzyme, was a protein. However, this ran against the authority of Richard Wilstard, who had the early isolated enzymes, in which no protein was detected. In fact, protein in Wilstard's samples had very small amount quantities. This was undetected by his techniques. Consequently, little attention was paid to Sumner's announcement that it was only when John Northrop succeeded in crystallizing further protein enzymes in the early 1930s that his work was properly acknowledged. What is urease? Urease is found in the natural environment such as water and soil and in human body, where its occurrence is connected with protein degradation. It's an enzyme and is capable of urine hydrolysis. One molecule of ammonia and one molecule of carbonate appear in the first step. Carbonate spontaneously converts into the second ammonia molecule and carbonic acid in a water solution, and ammonia is protonated. This urine hydrolysis results in pH increase. Urease and ammonia generated during the urine hydrolysis may be toxic for human tissues and probably have a role in long-lasting diseases like urinary tract infection or arthritis. Urease has many applications, such as urease can be used to analyze urine concentration in blood, urine, alcoholic drinks, natural water, and environmental wastewater. To analyze heavy metal content in natural water, wastewater, and soil. To control or shift the pH from multiple enzyme reaction system. To hydrolyze urine as sources of ammonia or carbon dioxide in special cases, and for the wastewater reclamation for life support system in space. Urease enzyme catalyzes the hydrolysis of urine, which occurs largely in the soils. Large number of bacteria and in soil process urease. A small group of bacteria known as urobacteria has an exceptional ability to decompose urine. Activity increases in proportion with the size of soil microbial population and organic matter contents. The presence of relatively fresh plant residue often results in abundant supply of urease. The greatest activity of urease is reported to occur in the rhizosphere, while microbiality is high and where it can be excreted from the plant roots. As Sumner said during his Nobel Prize lecture, we can sum up by saying that the result of discovering in the fields or the enzyme chemistry, some questions have been answered, but many new questions have arisen. We live in an expanding universe in more sense than that of the astronomers. I hope you feel in the same way. Thanks for watching this class.
大家好。萨姆纳17岁那年，在一次野外打猎活动中，因一个朋友不小心致使猎枪走火，恰好击中了他的左臂。医生切除了他肘关节以下的胳膊。这个不幸的事件曾给他沉重打击，但他不久便冷静下来，考虑自己的前途，决定正视现实，走自己的道路。他克服各种困难，用一只右手学会治理生活，又以坚韧的意志和毅力参加网球、滑雪、滑冰等体育活动，增强身体素质，而且用加倍的热情去学习各门功课。1946年，萨姆纳因提纯和分离出尿素酶结晶而荣获诺贝尔化学奖。身体的伤残和生理的缺陷可以折磨人的肉体和神经，但不能磨灭人的意志和为科学献身的精神。人生的支柱就是一颗坚强的心。好了，本次课到此结束，谢谢大家，再见。